How do you support a growing population? Well, the food chain tells us that mighty predators consume the prey beneath them, who then consume the prey beneath them, who continue eating each other until at the very bottom are plants. Plants are the basis of our nutrition and diets. Plants are the foundations of our ecosystems. Plants are what sustain life on our planet, and agriculture is the reason why humanity was able to form civilizations in the first place. It is no understatement to say that plants rule the world. But developing and breeding better crops to meet rising food demand is inefficient and slow. This is why it is more important than ever that we develop sustainable, effective, and long-term crop solutions to better support our growing population. I am a computational biologist. This means that I work at the intersection of mathematics, technology, and the life sciences. I am here representing the NC Plant Sciences Initiative, who is dedicated to answering the question, what if we could innovate our way to a lasting solution? I'd like to introduce you to a rising technology, 3D bioprinting. 3D bioprinting is just like the standard 3D printing that we are all familiar with, but with one key difference. Instead of printing with filament, we use living cells. We take these living cells and add them to a mixture of supporting enzymes and scaffolding materials to get bioink. We then take our bioink, load it into the printer, and then layer by layer, we are printing living tissue. Regenerative medicine has already made incredible strides using 3D bioprinting technology. We are building customized human organs in the lab to solve the donor crisis. In fact, the first cornea transplant using a 3D bioprinted organ was successfully completed by scientists out of Greensboro, North Carolina within the past year. But what does any of this have to do with plants? Every day, my team and I are working to address food demand challenges by developing a 3D bioprinting pipeline for full plant regeneration from single cells, from one cell to 1,000 plants. We source our cells from tissues with high regenerative capabilities, like the seed embryos. We then formulate the best bioink cocktails to sustain these cells and encourage them to grow and multiply. 3D bioprinting allows us to precisely arrange our bioink in 3 dimensions. By controlling cell density, spacing, and layering, we replicate the natural cues that tell our plants how to grow. Over months, we watch as single cells divide, form clusters, tissues, and eventually grow into full and healthy plantlets. But what can we accomplish with this? Well, while we are still in the early developmental stages of 3D bioprinting, it has already shown immense promise for agriculture. Have you ever gone into the supermarket and thought, that is a suspiciously perfect looking carrot? Well, there is a reason for that. It's estimated that upwards of 75% of food on supermarket shelves contain genetically engineered ingredients. Now consider the farms that grow these valuable crops. When floods or fires occur, the process to regrow and re-engineer these plants is costly, time-consuming, and labor-intensive. With 3D bioprinting, we can optimize these steps and have the potential to grow multitudes of plants in the lab until they're ready to put in the ground, without the risk of losing those genetic modifications. But this extends beyond modern-day crops. We are asking questions like, what if we could regenerate a plant found frozen in a glacier from tens of thousands of years ago? In this case, 3D bioprinting could be the key to reviving species which were originally thought to have been lost. What I want you to take away from today is that talented scientists every day are working to innovate lasting solutions. The future is calling us to explore 3D bioprinting, not just in regenerative medicine, but across disciplines. We are engineering our way through hunger, one seed at a time, because plants rule the world.